Welcome to your Biocast. I'm Mrs. Jaeger and I'll be your guide. As always, take detailed notes and don't forget to bring them to class the next day. Take some time right now to get your binder together, a pen, and get ready to learn more about biology. Welcome to your next Biocast video. This week's video is all about DNA, also known as deoxyribonucleic acid. You're already familiar with what DNA is, and you know that it codes for all of the genetic traits that you find in living organisms, but you're going to learn more about its structure today. Some cool facts about DNA. DNA stores information in an organism a lot like your hard drive does on a computer. Another cool thing about DNA is that human beings share 99.9% of their DNA with each other. So what that means is it's a very small percentage, just 0.1% of our DNA that makes us unique. You know that all living things have DNA, but I bet you didn't know that when you sit down to eat dinner, you're eating about 75,000 miles of DNA. So in the meat and the vegetables and the proteins that you're eating, within those cells, if we were to stretch the DNA out, it would reach about 75,000 miles. Kind of crazy. DNA can also be used to help assess your risk for certain genetic diseases, such as cystic fibrosis and even some types of cancer. Okay, moving on. DNA is made up of these pieces called nucleotides. Oops, sorry about that. So DNA is made up of these chunks called nucleotides, these molecules. And a nucleotide is made up of three things. It's made up of sugar, phosphate, and a nitrogen base. You do need to know that. There are four types of nitrogen bases. They go by C, A, T, and G, or cytosine, adenine, thymine, and guanine. We're going to learn how those pair up in a little bit. Okay, again with the nucleotide bases, they're divided up into two groups. The first groups are purines, and the second group are pyridamines. So nucleotides are divided into those two groups. You'll see that A and G, or adenine and guanine, are the purines, and cytosine and thymine are the pyridamines. Take a look at the structures that we have down here. Let's look at adenine. You've got this whole structure, which is adenine, okay? That's your nitrogen base. And attached to it, you'll see this thing. This thing is, if you see, it's labeled over here. It's a sugar, specifically deoxyribose. And then you've got this little molecule on the end, and that's a phosphate group. And you'll notice there's an open bond here. These bond together, and you'll see that in a later slide. So three things: base, sugar, and phosphate. Okay, so the structure of DNA. DNA is double-stranded. It's made up of two strands of those nucleotides that you just saw on the other slide. Okay, they're bonded together, and those bonds in between the nucleotide chains are called hydrogen bonds. We also see hydrogen bonds in water. The sugars and phosphates make up what we call the backbone. If you think about your backbone, it's kind of the structure that holds everything together in your body. The sugars and phosphates also make up the backbone in the DNA. And here is a model of what the full structure of DNA looks like. You'll notice that you see those two strands, right? We've got one strand here, we've got another strand here. You have these bonds in between the nucleotide bases. These are the hydrogen bonds. You've got your whole nucleotide with your sugar and your phosphate and your nitrogen base. And they color code them nicely. We'll notice how they pair up. We'll go through that in a little bit. Okay, DNA was discovered by a group of scientists working together. Actually, these two worked together. Oops. These two gentlemen here worked together, and this was a woman who discovered DNA, and pretty much these guys took her research and really didn't give her credit for her work. She didn't get credit till later in life. It's kind of unfortunate. She used X-rays to study the structure of DNA, and she discovered this helix or twisted ladder shape. Okay. Watson and Crick used that X-ray to build a more accurate model, and the model that we accept today is called the double helix. You could also think of it as a twisted ladder, because that's what it looks like. Okay, the way that the nucleotide bases pair is consistent. There are rules that we follow. Adenine pairs with thymine, and cytosine pairs with guanine. So if you can think of A and T pairing together, spelling the word at, and I like to think of C and G bonding together. They're kind of the same shape of letter, C and G, and those are the ones that pair together. A's pairs with T's, C's pairs with G's. So look at how we have it here. A's pair with T's. Sorry about that, guys. A's pair with T's, and you see C's pairing with G's. Okay, again we have G's pairing with C's, A's pairing with T's, right there. So how about doing a little practice? What I want you to do now is write down this strand right here. Notice how they're grouped into threes. We'll talk about why a little bit later. And I want you to write the complementary strand. So the strand that's going to go here is called the complementary strand. Okay, so see if you can do that. Let's see, A is going to pair with what? T. Good. T pairs with A. G pairs with C. See how those are complementary? C pairs with G. A pairs with T. C pairs with G. Okay, see if you can finish the other ones on your own. And I will see you in class when you bring these notes and are ready for your little mini quiz. Hope you enjoyed this biocast on the structure of DNA. We're going to learn more about our RNA in our next biocast.